Okay, for today's lab, we are doing standard qualitative analysis of water. So for the first sample that we will use is from the kolam kejuruteraan. So now we are transferring 10 mole of the sample from kolam fakulti kejuruteraan into the double strength lactose fermentation broth. For the second round, we will transfer 1 mole of the same sample from kolam fakulti kejuruteraan into the 5 single strength lactose fermentation broth. We will be repeating the same procedure for the 3 remaining single strand lactose fermentation broth. For the third round, we are now transferring 0.1 mole of the same sample from faculty kejuruteraan into the single strand lactose fermentation broth, and then we will repeat the same procedure for the 4 remaining lactose fermentation broth. For the second sample, we'll be using the sample from the swimming pool of UPM. So we are now transferring 10 mole of the sample from the swimming pool UPM into the 5 double strength lactose fermentation broth. For the next round, we will be transferring 1 mole of the water sample from the swimming pool of UPM into the 5 single strength lactose fermentation broth. And then we will be repeating the same procedures for the 4 remaining single strength lactose fermentation broth. For the last round, we will be transferring 0.1 mole of the water sample from swimming pool of UPM into the 5 single strength lactose fermentation broth. And then we will be repeating the same procedures for the 4 remaining single strength lactose fermentation broth. We have now finished transferring all the water sample from the pool of Fakulti Kejuruteraan and also from the swimming pool into all the lactose fermentation broth, and all this will be incubated for 48 hours at 37 degrees Celsius. Okay, so we will now proceed to the confirm test for the standard qualitative analysis of water. After the 24 hours of incubation, we have now observed that the water sample from the Fakulti Kejuruteraan contains the presence of gas bubble. So we will take out one bottle of lactose fermentation broth, which is the 0.1 mole of lactose fermentation broth, to proceed into the confirm test to do the strict plate for EMB agar plate. The strict plate on EMB agar plate is now completed. So for the water sample from the swimming pool of UPM, we will further the incubation period for 48 hours. So after the incubation period of 24 hours of the water sample from swimming pool UPM, we can clearly see that there is no water bubble, there's no air bubble presence in the lactose fermentation broth. So we can conclude that the water sample itself is clean and not contaminated. So now we will proceed with the water sample from Kolam Fakulti Kejuruteraan. We will inoculate the sample from Kolam Fakulti Kejuruteraan into the agar slant. And then we will also inoculate the sample into the lactose fermentation broth. So the inoculation is now completed, and then we will incubate all the plates culture in an inverted position for 24 hours at about 37 degrees Celsius. So after the incubation period of 24 hours for the water sample from Kolam Fakulti Kejuruteraan, we can now observe that there are some air bubble presence in the lactose fermentation broth. Also, we can see that there are some presence of bacteria colony in the agar slant. So now we will perform the grain staining for the bacteria colony from the agar slant. So we will now streak up some bacteria colony from the agar slant. After streaking out the bacteria colony from the agar slant, we will smear it on the cover slide. We will smear it on the slide. And then we will cover the smear with crystal violets for 30 seconds. So we will now wait for 30 seconds. Now we will gently wash off the crystal violets with distilled water. We will cover the smear with iodine for 10 seconds. Okay, 10 seconds has completed. We will now wash it off with distilled water. And then we will decolorize it with ethanol. And then we will cover the smear with safranin for 30 seconds. So now we will wash it off with distilled water. And then we will gently tap the cover slide with some tissue paper to dry it. So the grain staining is now completed. So after the grain staining is completed, we will now put some drops of immersion oil onto the cover slide and then cover it. Okay, so this cover slide will be used to be observed under microscope.